Hi and welcome to my OCR A A-Level Biology revision session with me, Christine. So today's lesson I want to look at PCR, gel electrophoresis and DNA profiling. So first of all, let's just discuss the genome. All the genetic material of an organism is known as their genome. Now, if we're looking at eukaryotes, we're therefore talking about their nuclear DNA, the mitochondrial DNA, and if we're talking about plants with eukaryotes, then we also need to include the DNA which is found in the chloroplast. So the genome is all the genetic material of the organism. Now, the human genome is approximately 3.2 billion nucleotides long. And the chromosomes we know are made up of millions of DNA base pairs. Now, only 2% of your total DNA are genes, which are known as exons, which code for proteins. So because we know this, and because we have sequenced the entire base sequence of the human genes, which is known as the Human Genome Project, because this has been sequenced, we've learned a lot more about what those sequences of DNA bases actually relate to with regards to proteins. And so we're going to learn more about this with regards to DNA sequencing in a whole other video. But what I want to focus on in today's lesson is about PCR and DNA profiling. And for that, we need to understand about satellite DNA. So satellite DNA are short sequences of DNA which are repeated many times. Now, they are found within your introns. So remember, your DNA, you have your coding region, which is transcribed into pre-mRNA. That consists of your exons and introns. And we know through the process of splicing that we end up with our mature RNA, but those introns have been removed. So those introns that were removed, they are repeated sequences of this satellite DNA. Also, your telomeres, which are found at the end of your chromosomes, and the centromeres that hold the sister chromatids together, are also areas where we can identify these satellite DNA. So you can have what's known as a microsatellite, which is a short tandem repeat. It's about 2 to 4 base pairs which are repeated about 5 to 15 times. So in this case, my microsatellite is the combination AGAT. So that's 4 base pairs which are going to be repeated, and that can be anything between 5 to 15 times. So in my diagram, we have got 4 of them that we can see in the repeated sequence. Now, a mini satellite is what's also known as your variable number tandem repeats. Now, that can be between 20 to 40 base pairs which are repeated, and they can be repeated 50 to several hundred times. So it's important we know that this satellite DNA is talking about those microsatellites and these mini satellites. Now, if they're going to go into detail in the exam, they'll give you some information, i.e. they'll call them variable number tandem repeats. But what you need to understand is that these satellites always appear in the same positions on the chromosomes, but the number of repeats for each of those mini or microsatellites varies between individuals. Therefore, different lengths are actually inherited from both parents. So the more closely related individuals are, the likelihood will be that they will have similar satellite patterns but not identical. Therefore, what we can do is we can produce an image of the patterns in the DNA of an individual, and that is what's known as DNA profiling. So let's look at how we can do this DNA profiling. So the first thing we would need to do is extract a sample of the DNA from their cells. And we know that that DNA is held in the nucleus and also in the mitochondrion and if we're looking at plants in the chloroplast. So we would extract samples of the DNA. Now what we need to use are what's known as restriction endonucleases. Now these are enzymes that are going to digest the DNA strands into varying length fragments. They're going to cut at very specific recognition sites. So you can use different types of restriction endonuclease. So if I use the EcoRI, what's going to happen is that will look for recognition sites and that will cut wherever it sees them. So this would therefore produce 3 fragments in my DNA strand. If I used a different restriction endonuclease, that would cut at a specific recognition site. That would therefore leave me with 2 fragments. If I'm looking at the same strand of DNA as my original strand, these 2 different restriction endonucleases are going to cut at different sites, therefore giving me different fragments. And if I were to put the 2 of them together, again, that would therefore result in me getting different fragments because they will cut at specific recognition sites at exactly the same recognition sites. So once we have cut our DNA, we've taken our sample, we've cut it into fragments, what we would then do is what's known as PCR, polymerase chain reaction. So a PCR is a machine that's going to be used to copy and amplify minute quantities of DNA. So we do this through a process of first heating the temperature up to 96 degrees C. Now that thermal energy that we're putting into the fragments is going to cause denaturation. It's going to break the hydrogen bonds between those base pairs. We also then will add into our sample a DNA primer. Now, a DNA primer is a short sequence of DNA complementary to the single-stranded template, and that will anneal. Now, annealing is basically where it's going to form hydrogen bonds between the complementary base pairs, and it will form it in that sequence. So we've dropped the temperature down. We're now annealing this primer to position. And then what we're going to do is we're then going to extend, because the key thing to note is we need a primer because DNA polymerase cannot form phosphodiester bonds off of nothing. So the primer needs to be there so that we can then extend to make these fragments. So what we do is we use a Taq polymerase. Now this is a polymerase that comes from a prokaryote that can withstand higher temperatures. So we've raised the temperature back up to 60 degrees C and we extend. So what we're doing is we are building these phosphodiester bonds on this strand, and the new free nucleotides which are within our sample will be able to form complementary base pairing, forming hydrogen bonds wherever there is a complementary base. So as long as we have enough free nucleotides, we have our primer, we have our Taq polymerase, and our restriction endonuclease is present, we can do the process of PCR, this amplifying. So the whole point in the polymerase chain reaction is that we have our original DNA, we have free nucleotides, a primer, and our Taq DNA polymerase in our DNA sample. The PCR machine is then going to go through a cyclic process of denaturation, annealing, extending. So the first time it does that, the first cycle, we're going to go from having our original DNA and we are then going to end up with 2 copies. 2 copies that are semi-conservative, 1 original strand and 1 new strand in the DNA molecule. So therefore we end up with 2 copies. Now if we go through a second cycle, we're obviously going to end up with 4 copies. If we go through a third cycle, we're going to end up with 16 copies. And if we go through a fourth cycle, we're going to end up with 32 copies. Therefore, what we have to understand is that what we are doing is we are always looking at how many cycles it is going through. So how many cycles are we going through? We are going through, in one process of using the PCR machine, about 30 cycles. So this will be repeated many times to the point in which we will end up with almost 2 billion copies of the DNA, the original sample that we took. Now it's important to note that to figure this out, we take the final number of copies is our initial copy, 1 in this case, times 2 to the power of the number of cycles that you have done. So if we've got 30 cycles, we therefore would do 1 times 2 to the power of 30. Well, what if we have 10 copies? It's the same process. We have 10 times 2 to the power of 30, so therefore we would end up with over 10 billion copies of our sample. So it's important to note that whenever you are doing this, you need to understand that the whole process of a polymerase chain reaction is that we are amplifying this minute original DNA copy. So then what do we do with it? Well, the next thing we need to do is what's known as gel electrophoresis. So we can separate out proteins and DNA fragments by using gel electrophoresis. So we take our wells, which are made up of agarose jelly, and these are gaps in the agarose jelly, and we place that into a buffer solution. And then what we do is we put our DNA samples inside the well. Once we've done that, we then expose these fragments to an electric current in that trough of agarose gel. And we need to remember that our DNA is negatively charged. So we need to make sure that we place the wells with the DNA samples at the side where the cathode is going to go. Because what's going to happen is those fragments are going to be repelled from the cathode and they are going to move towards the anode, and they will move at different rates through the gel. The smaller the fragment, the faster it will move. And what will happen is our DNA will separate into bands according to the size of the fragments. Now it could be proteins that we're putting in there, and they would work in the same way, that they would separate out based on, one, their charge. We'd have to make sure that their charge was not affecting the rate at which they move through. We only want them to be separated based on their size. So we would need to ensure that we are affecting it looking at the size of the fragments. So what would we then do? Well, we want to visualize them, and the only way we can visualize them is by doing what's known as hybridization. So the first thing we do is we take our gel and we need to immerse it in an alkaline solution. Now this is going to cause denaturation, and that will therefore break those hydrogen bonds between our double-stranded DNA, and it will leave us with single-stranded DNA fragments. We will then do a process called southern blotting. What we're going to do is we're going to transfer the fragments to a nylon membrane using capillary action. We're going to put layers of the material which is going to pull through the alkaline solution with the DNA fragments, which will therefore transfer them onto this nylon membrane. The DNA fragments can't get through the nylon membrane, only the alkaline solution can, and therefore that will cause them to be transferred to that nylon membrane. What we then do is we put radioactive or fluorescent DNA probes. Now these DNA probes are short DNA or RNA sequences which are complementary to our microsatellites. Now this is through that process known as hybridization. Because we have these short DNA and RNA sequences which will complement those microsatellites through forming hydrogen bonds, we can therefore tag them with these radioactive or fluorescent DNA probes. and then we will then expose them to either X-rays or ultraviolet light, and that will therefore make the bands visible. So the picture that I'm showing in this diagram was where we used ultraviolet light, and we had a very specific camera that would take pictures of our DNA fragments that we had separated out. The one that's going all the way up to the top, that is what's known as a DNA ladder. So when we use restriction endonucleases, we can use them to digest the fragments into smaller fragments, cutting at specific recognition sites. So what we can do is we can put the sample with only EcoRI, we can put it in with only the BAM-HI, and we can put it in with both of those restriction endonucleases. And because we have a DNA ladder of known DNA-based sequences, we can see how they will separate out into bands. So if we start with the first one then, when we use only the EcoRI endonuclease, it separates and gives us 3 fragments, a really small fragment which is 2 kilobases. We have a slightly bigger one of 4 kilobases, and then we have one which is 10. By using the BAM-HI, what we have is that will separate them into 2, but this time they are 8 kilobase pairs. Now, if you look at the size of the band here, you can see that the band is thicker, therefore indicating that there are 2 of the same length there. So the bands will actually give you an indication of how many of them are there based on the size of the band. And if we obviously include both of the restriction endonucleases together, what we can see is that we get a 6 kilobase pair, 2 4 kilobase pairs, and a 2 kilobase pair. So the banding, the way they separate out, allows us to identify how they can be similar through looking at those patterns. So DNA profiling is actually used in forensic science, where we'll take a DNA sample, i.e. blood that's left, semen, saliva, hair roots, or skin cells, and then we will use PCR to amplify that DNA sample. We will then use restriction endonuclease and gel electrophoresis to separate them out into their fragment sizes, and then hybridization, and then compare that to the suspect's DNA or to a criminal DNA database where we have that detail stored. Now, unfortunately, forensic science is only as good as the scientists who are collecting the evidence, the lab technicians who are using it, and the data that's being stored in the database. So it's important to note that sometimes there can be mistakes made, and that can either destroy a criminal case or can help solidify the evidence for a criminal case. So you have to be aware that we can use it in forensic science, but it can also cause some problems if it's not done correctly. We can also use DNA profiling in the analysis of disease risks. So we know that non-coding microsatellites have these repeating patterns, and we've actually found that they can be associated with an increased risk of certain diseases like cancer or heart disease. So we can do DNA profiling to identify whether or not someone is at risk of a certain disease. But we will look at that in more detail when we look at DNA sequencing in my next video. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. And if you haven't already done so, please do check out my revision platform, www.aiqchat.com.